Welcome, everyone. We're diving deep into HIV vaccine research today, exploring how far we've come and where it's all heading. Especially relevant, I think, for our listeners here on the HIV RNA podcast, our main source. A scientific review paper just came out last February. Get ready to be surprised. The length of this search, the variety of approaches, even unexpected factors, all playing a part in future vaccine development.
Truly a fascinating journey. You know, the search for an HIV vaccine started almost 40 years ago. Can you believe that? 4 decades, scientists trying to crack this puzzle.
40 years? That's longer than I've been alive. It really puts into perspective just how complex HIV is and how persistent these researchers have been. Wow.
It does indeed. And the approaches, they've evolved over time. Early focus was antibodies. The thinking was if you could get the body producing enough, they'd neutralize the virus before it even infected cells.
It's pretty straightforward. So what happened? Why didn't that work out?
Well, HIV is a master of disguise. It mutates rapidly, meaning it can change its appearance, evade those antibodies through a major wrench in those early efforts.
So they had to switch gears then.
Exactly. They started focusing on CD8 plus T cells, kind of like the special forces of your immune system. These cells can directly kill cells that are already infected with HIV.
Ah, so different strategy then. Instead of preventing infection, it's about controlling it once it's there.
Precisely. And for a while, this seemed really promising because those T cells could respond to a variety of HIV strains. However, even though these T cell vaccines generated strong immune responses, they couldn't always prevent HIV acquisition.
So back to the drawing board.
In a way, yes. But each of these research avenues, even the ones that didn't lead to a licensed vaccine, provided valuable insights, like solving a complex puzzle, each attempt revealing a new piece.
So tell me more about these different vaccine approaches. I know the paper dives into protein vaccines, vector vaccines, and something called broadly neutralizing antibodies. What are those exactly?
Let's start with protein vaccines. They use pieces of the HIV virus itself to trigger an immune response. Think of it like showing the immune system a mugshot, so it can recognize and attack it.
Okay, makes sense. And what about vector vaccines? Those sound a little bit more complicated.
They are a bit more complex, yes. Vector vaccines use harmless viruses, like a common cold virus, to deliver HIV genes into our cells, like sending in a Trojan horse, with instructions on how to fight HIV.
Wow, clever strategy. And then there are these broadly neutralizing antibodies. Sounds like something out of a superhero comic. What makes them so special?
They're called broadly neutralizing because they can target a wide range of HIV strains, even those that have mutated. Most antibodies, very specific, like a key that only fits one lock. But broadly neutralizing antibodies, more like a master key, can open many different locks.
Ah, I see. So they could overcome HIV's ability to change its appearance and evade the immune system. That's why they're considered so promising.
Exactly, like the elite force of the antibody world.
So the researchers have been exploring all these different approaches. But it sounds like it's been a bit of a roller coaster ride, with both successes and setbacks.
That's a great way to put it. Let's look at some specific trials, understand this roller coaster ride a little better.
Okay, I'm ready for the ride. Where do we start?
Well, some of the early trials, like VAX-003 and VAX-004, used those protein-based vaccines we talked about. Unfortunately, they didn't achieve the desired level of protection.
So a setback right out of the gate.
You could say that. However, even though they didn't work as hoped, these trials were crucial. They shifted the focus away from just generating any antibody response to generating the right antibody response. They highlighted the need for antibodies that could target the most vulnerable parts of the HIV virus.
So a valuable lesson learned, even if it wasn't the outcome they were hoping for. What about the other approaches? What happened with the vector vaccines?
There were high hopes for the vector vaccines, especially in trials like STEP and HVTN 505. These used an adenovirus vector to deliver the HIV genes, but they ran into some unexpected safety concerns.
Uh-oh. What kind of safety concerns?
It turned out that people who already had immunity to adenovirus from previous exposure, perhaps through a common cold, had an increased risk of HIV infection after receiving the vaccine.
Wait, so the vaccine was actually making them more susceptible to HIV? That's pretty scary.
It was a major setback, yes. But it also highlighted the importance of understanding pre-existing immunity and how it can influence vaccine responses.
That makes sense. Sounds like every step forward in HIV vaccine research also uncovers new complexities.
You're absolutely right. And the story doesn't end there. There was another trial, HVTN 702, based on the modest success of an earlier trial called RV144. RV144 showed about 31% efficacy. Not great, but it was the first time a vaccine had shown any protection against HIV at all.
Okay, so there was a glimmer of hope there.
There was. And they adapted the RV144 vaccine for a different HIV subtype, for the HVTN702 trial. But sadly, it didn't show any significant protection.
Another disappointment. What about those broadly neutralizing antibodies? Did they have any better luck?
The AMP trials focused specifically on broadly neutralizing antibodies, and they did show some potential. They demonstrated that these antibodies could prevent HIV infection in some cases. However, they also highlighted the need for combination approaches to truly tackle HIV's incredible diversity.
So even the super antibodies aren't a silver bullet.
Unfortunately not.
Yeah.
Seems that HIV always has another trick up its sleeve. But the AMP trials were a major step forward because they proved that broadly neutralizing antibodies could work. Now it's a matter of figuring out how to make them even more effective and durable.
It's incredible how much work has gone into this. And it sounds like even the setbacks have taught researchers valuable lessons.
Absolutely. Every trial, successful or not, has contributed to our understanding of the virus and the immune response.
So we've talked about antibodies, T cells, different types of vaccines, and these amazing super antibodies. What else is in this research paper? Where do we go from here on this roller coaster ride?
Well, before we jump into human trials, there's a crucial step. That involves our primate cousins.
You're talking about animal testing.
Yes, but with a specific type of monkey called a rhesus macaque.
Why macaques? What makes them so special?
Rhesus macaques are susceptible to SIV, simian immunodeficiency virus, very similar to HIV. Causes a similar disease in monkeys.
So it's like a model for studying HIV in humans.
Exactly. By testing potential vaccines in macaques first, researchers can get a better understanding of how the vaccine might work in humans and whether it's safe to move forward with human trials.
Makes sense. It's like a stepping stone to ensure the safety and efficacy of the vaccine before it reaches people.
Precisely. And over the years, researchers have made interesting discoveries about how to use macaques to get the most accurate information about potential HIV vaccines. One of the key things they've learned is that the challenge dose matters.
Challenge dose? What's that?
It refers to the amount of virus the macaques are exposed to during these trials. In the past, they often use high doses of SIV, led to rapid infection in the animals.
But that doesn't really reflect how HIV is transmitted in the real world, right?
You're right. HIV transmission usually happens through exposure to much smaller amounts of the virus. So now researchers are shifting towards using low dose, repeated challenges in macaques.
So they're trying to mimic real-world HIV transmission as closely as possible.
Exactly. This helps them get a more accurate picture of how well a vaccine might work in preventing HIV infection in humans.
It's amazing how much they've learned about how to design these trials to get the most relevant information.
It really is. And this leads us to one of the most fascinating aspects of recent HIV vaccine research, the realization that there's more to the story than just antibodies and T cells.
Okay, now I'm really intrigued. What else is there?
We're going to delve into the world of innate immunity and pre-existing immunity. These are factors we're only beginning to understand, but they could hold the key to developing a truly effective HIV vaccine.
So, going beyond the usual suspects and exploring new territory, this is where things get really interesting. I can't wait to hear more about this in the next part of our deep dive.
You know, picking up on that idea of pre-existing immunity, it's a bit of a double-edged sword. On one hand, can cause problems, like we saw with those adenovirus sector trials, but on the other hand, it could offer some unexpected advantages.
Okay, now I'm really curious. Tell me more about this pre-existing immunity thing. How could it be beneficial when it comes to HIV vaccines?
Well, the research paper highlights a really intriguing example, the BCG vaccine for tuberculosis.
You mean that vaccine that's been around for like 100 years? What does that have to do with HIV?
It's fascinating, right? The BCG vaccine has been around for over a century, and we're still discovering new things about it. There's growing evidence that it might offer protection beyond just tuberculosis. For example, studies have shown that BCG vaccination can reduce the risk of other infections in infants, including respiratory infections and even sepsis.
Wow, I had no idea. So it was like a multi-purpose vaccine?
In a way, yes. And it gets even more interesting when we look at its potential impact on HIV. Epidemiological studies have suggested a link between BCG vaccination and a lower risk of HIV infection.
Really? So people who have had the BCG vaccine might be less likely to get HIV?
That's what the data suggests, though it's important to note that it's not a direct, foolproof correlation, but it's a fascinating observation nonetheless. And it's spurred a lot of research into why this might be happening.
I can see why that would be intriguing. What's the leading theory? Is it actually like boosting the immune system somehow.
One of the hypotheses is that BCG might be inducing something called trained immunity. Relatively new concept in immunology, but it's gaining a lot of attention.
Trained immunity. Okay, that sounds intense. What exactly does that mean?
Essentially, it means that the immune system, after encountering a pathogen, like the one in the BCG vaccine, develops a kind of memory, allows it to respond more effectively to a wider range of threats, even those unrelated to the original pathogen.
So it's like giving your immune system a workout, so it's ready to tackle anything that comes its way.
That's a great way to put it. Boot camp for your immune cells. And this concept of trained immunity has become a hot topic in HIV vaccine research. If we can figure out how to harness it effectively, could be a game changer.
So BCG is showing some promise. But what about other examples of pre-existing immunity? I know the paper also mentioned CMV, right?
Yes, cytomegalovirus, or CMV, another interesting case. Very common virus. Most people are infected with it at some point in their lives, usually without any noticeable symptoms. But what's fascinating is that CMV has a profound impact on the immune system, even if you don't realize you've been infected.
In what way? What does it do?
It can actually reshape the immune landscape, leading to the expansion of certain types of immune cells, particularly natural killer cells.
Or NK cells for...
NK cells, what are those? NK cells are like the assassins of the immune system. They're constantly on patrol, looking for cells that are infected or cancerous, and they eliminate them without mercy.
Wow, sounds pretty intense. So having CMV could actually make your immune system more powerful.
It's not quite that simple. There's research suggesting that CMV infection could enhance the immune response to other viruses. But the relationship between CMV and HIV vaccine efficacy is complex and not fully understood.
So it could be a good thing or a bad thing, depending on the circumstances.
Exactly. Like having a powerful weapon that can be used for good or for evil, all depends on how it's wielded.
Seems like every time we uncover a new layer of this HIV puzzle, it just gets more and more complicated. But that's what makes it so fascinating, right?
Absolutely. Like peeling an onion. But instead of tears, we get insights into the incredible complexity of the immune system.
So we've talked about BCG and CMV as examples of pre-existing immunity. But what does this all mean for the future of HIV vaccine research?
Well, it opens up a whole new avenue of exploration. If we can understand how pre-existing immunity influences the response to HIV vaccines, we can potentially design vaccines that work better for different populations.
So a one-size-fits-all approach might not be the best strategy.
Exactly. We might need to tailor vaccines based on an individual's immune history, their previous exposures to other pathogens, and their unique immune profile.
That sounds incredibly challenging.
It is, but it's also incredibly exciting. We're moving towards a more personalized approach to vaccination, where we can potentially predict how someone will respond to a vaccine based on their individual characteristics.
So it's like moving from a mass-produced vaccine to a custom-made vaccine.
You could say that. And this brings us to another exciting development in HIV vaccine research, the use of mRNA vaccines.
Oh, yeah. Those were all over the news during the COVID-19 pandemic. They were incredibly effective.
They were, and now scientists are exploring their potential for HIV as well. mRNA vaccines have a lot of advantages. They're relatively easy to manufacture and modify, makes them very versatile.
And how do they work exactly? I remember hearing something about genetic code, but it was all a bit over my head at the time.
It's actually pretty straightforward. mRNA vaccines work by delivering a piece of genetic code from the virus directly into our cells. This code instructs our cells to produce a viral protein, which then triggers an immune response.
So it's like giving our cells a recipe to make a piece of the virus, so our immune system can learn how to recognize and attack it.
That's a great analogy. And because mRNA vaccines can be easily modified, they can be designed to target specific parts of the HIV virus, potentially making them more effective than traditional vaccines.
This all sounds very promising. Are there any mRNA HIV vaccines in development right now?
There are. In fact, there are several trials underway, testing mRNA vaccines for both HIV prevention and treatment. Still early days, but the initial results are encouraging.
That's fantastic news. Sounds like we're entering a new era of HIV vaccine research.
Yeah.
One that's full of potential and new possibilities.
I agree. There's a lot of excitement in the field right now. And another area that's generating a lot of buzz is structure-based vaccine design.
Okay, now you've lost me again. Structure-based vaccine design. What in the world is that?
It sounds complicated, but it's actually quite elegant. It involves using our knowledge of the three-dimensional structure of the HIV virus to design vaccines that are more likely to trigger a strong and effective immune response.
So it's like building a vaccine based on the virus's blueprint.
Precisely. Scientists are using this knowledge to design stabilized versions of the HIV envelope protein, which is the part of the virus that attaches to our cells.
Why do they need to stabilize it?
Well, the HIV envelope protein is very good at changing its shape, makes it difficult for the immune system to recognize and target.
Ah, so stabilizing it makes it easier for the immune system to lock onto it.
Exactly, like presenting the immune system with a clear target, rather than a moving target.
I'm starting to understand. So by understanding the structure of the virus, they can design vaccines that are more precise and effective.
That's the idea. And some of these structure-based vaccine candidates have shown promising results in preclinical trials.
This is all so incredible. It really feels like we're on the cusp of a breakthrough. But I'm also realizing just how complex this whole field is, and how much work still needs to be done.
You're right, marathon, not a sprint. But the progress that's been made in recent years is truly remarkable. We've come a long way since those early trials, and there's every reason to be optimistic about the future.
I'm definitely feeling optimistic after learning about all these new approaches. But before we get too carried away, I'm sure there are some challenges and ethical considerations that we need to address as well. And I'm also curious to know what all of this means for people listening to the HIV RNA Test Guide podcast. What practical implications might these new vaccines have for them?
Those are excellent questions, and we'll explore them in more detail in the next part of our deep dive.
All right, back for the final part of our deep dives into HIV vaccine progress. My brain is definitely full after all that.
It is a lot to take in, but it's also incredibly exciting to see how far the research has come.
It really is. Before we wrap up, though, I wanted to touch on something I mentioned earlier. What does all this research mean? For people listening to the HIV RNA Test Guide podcast, any practical implications for them?
That's a great question. It's important to remember that even though there's no licensed HIV vaccine available just yet, there's still a lot of progress being made. The research we've been discussing today offers hope for the future.
Hope for a future where HIV is no longer a threat. That's definitely something worth striving for.
Absolutely. And while we wait for that day to come, it's important to stay informed about the latest developments in HIV prevention, testing, and treatment. That's where resources like the HIV RNA Test Guide podcast come in.
Exactly. Knowledge is power. Understanding the tools available to us can make a real difference in our lives and the lives of those around us.
I couldn't agree more.
So as we wrap up this deep dive, I'd like to shift gears a bit and talk about some of the broader implications of this research. What stands out to you most from this review paper?
What fascinates me is how this research is pushing the boundaries of our understanding of the immune system as a whole. It's not just about finding an HIV vaccine, it's about deciphering the intricate language of immunity. We're learning how our bodies interact with pathogens, how we defend ourselves, and how we can potentially manipulate these processes to our advantage.
It's like we're unlocking the secrets of our own bodies.
Exactly. And these insights have implications far beyond just HIV. The knowledge gained from studying this incredibly complex virus is informing vaccine development for other diseases, even cancer. We're learning how to train the immune system to be more effective, more precise in its attacks.
So the ripple effects of this research could be enormous, like a rising tide that lifts all boats.
That's a great analogy. And the paper also touches on something I think is incredibly important to acknowledge. We've talked about all these amazing scientific advances, but we can't forget that this research involves a lot of trial and error, and sometimes things don't go as planned.
You're talking about the setbacks, the trials that didn't work?
Yes, and it's important to remember that even those setbacks provide valuable information. They help us refine our understanding, guide us towards more effective solutions. Process of constant learning and adaptation.
It's a reminder that science is a journey, not a destination.
Well said. And it's a journey that requires patience, perseverance, and a willingness to embrace both successes and failures.
Speaking of failures, were there any specific challenges or roadblocks highlighted in the paper that really struck you?
One of the biggest challenges, and we've touched on this throughout our conversation, is HIV's incredible diversity. This virus is constantly changing, mutating, evolving. It's like a moving target. And that makes it incredibly difficult to design a vaccine that can effectively neutralize all the different strains.
So it's like trying to hit a bullseye on a dartboard that's spinning wildly.
That's a great way to put it. And that's why researchers are exploring combination approaches, trying to target multiple parts of the virus, or using multiple strategies to stimulate the immune system.
Sounds like they're pulling out all the stops.
And they are. And it's not just about designing the right vaccine, it's also about delivering it effectively. We've talked about different vaccine platforms, but there's also the challenge of ensuring that the vaccine reaches the right cells and triggers a durable immune response.
So the multifaceted challenge, to say the least.
It is. But I think it's important to end on a note of optimism. Despite all these challenges, the progress that's been made in HIV vaccine research is truly remarkable, and the future holds immense potential.
I'm definitely feeling hopeful after this deep dive, and I think it's safe to say that the quest for an HIV vaccine is in good hands.
I wholeheartedly agree.
Well, on that note, I think we've covered a lot of ground today, from the history of HIV vaccine research to the latest breakthroughs and the challenges that lie ahead. It's been a whirlwind of information.
It has, and I hope our listeners have gained a deeper appreciation for the complexity of this field, the dedication of the researchers, and the incredible potential that lies ahead.
I couldn't have said it better myself. So, to all of you listening, thank you for joining us on this deep dive. Stay curious, stay informed, and stay hopeful.
We'll see you next time on The Deep Dive.